All right, human anatomy, we're going to go over everything you should know for the adaptive immune system. Okay, so we're going to start here, work our way down, and we're going to get right into it. So the adaptive immune system is the third line of defense after the innate immune system. So after you go through the skin, after you go through all the internal defenses of the innate immune system that we talked about, you activate the adaptive. All right, so there's two types of immunity that the adaptive immune system has. You have humoral immunity, which is when the antibodies float through the humors of the body that seek out those extracellular antigens to be trained and to learn and to ultimately attack and defend your body against foreign pathogens. And you have your cellular immunity, which is where self cells attack foreign or cancer cells directly. So these are your own cells, they're your own T cells attacking these cells directly to defend against foreign pathogens. So let's start out. What is an antigen? Okay, an antigen is an extracellular protein that triggers the adaptive defenses of the body. A complete antigen will have immunogenicity and reactivity. Immunogenicity is the ability to stimulate the lymphocytes to divide, to proliferate, to do clonal selection, and to divide to ultimately create a stronger, more powerful response to fight foreign bacteria. And the reactivity is the ability to react to the activated lymphocytes and antibodies to do things like release histamine, recruit more leukocytes to that area. Now a hapten is, think of it as a half antigen. Okay, a hapten is reactive, but it does not have the ability to stimulate lymphocytes to proliferate, so it's only doing this. Examples of that include things like poison ivy, penicillin, and detergents. So now we're going to get into lymphocyte T and B maturation. So the lymphocyte maturation, we have our T cell and we have our B cell. All of the stem cell for both of them is going to be in the red bone marrow. The T cells are going to then soon move to the thymus to maturate, and the B cells are going to remain in the red bone marrow to do maturation. Now these steps of maturation are the same, but they're just happening in two different locations for the T and the B cell. So again, they both originate in the bone marrow. The T cell moves to the thymus and the red bone marrow. The T cell moves to the thymus. Again, B cell stays in the red bone marrow. And at stage 2, the T and the B cells, they are learning immunogenicity. Okay, immunogenicity is the ability to, well, basically what immunogenicity means is that these cells are becoming educated and they're learning that they need to attack an antigen. They just don't know what it is yet, but they're being trained and they're being educated on, okay, we need to be able to attack antigens. In stage 3, this is where the immune cells, so the naive, never having been exposed to antigen, T and B cells, so they're not exposed yet, but they're about to be, because they're about to enter lymphoid organs. Specifically, the lymph nodes and the spleen. In the lymph nodes and the spleen, this is where they will encounter their antigens that they have been educated on in step 2. I should also note that in step 2, the T and the B cells learn self-recognition, so they learn that they should not attack their own body's cells. If they do attack their own body's cells, then you get things like autoimmune condition. But back to step 4. So once they're educated about the lymphocyte they need to attack, an antigen will select it for clonal selection. So that means proliferation to make more B cells or to make more T cells. So they're going to proliferate to make more cells that can recognize that antigen. Now moving on over here, everything from this line over, this is all background knowledge that you should know. And now there's actually a little bit more background knowledge I want you to know. So come on over here, follow me. If we look at what a antibody is and how it's structured, we have a heavy chain variable region, which is going to be located here. Heavy chain constant region located here. I have a light chain variable region located here in the light chain, and also a light chain constant region located right here. So this is just a little bit more of the anatomy of how antibodies are structured. Now, there's 5 classes of antibodies that you should be aware of, all right? Starting out, we have the IgM antibodies, which are secreted by plasma cells during the primary response. So this is during the first response is when these IgMs are secreted, and they activate the complement pathway. The IgGs are released during the secondary response of the immune pathway, where they will also activate the complement. And we're going to go over how we can see that practically in just a second. You can think of IgGs as the secondary response because they're the most abundant. There's the most of them. So think of like an Instagram person who wants to make the most amount of Instagram posts. IgG. All right, now we move on to the IgAs. And these IgAs are found in our saliva, sweat, intestinal juice, and milk from a mother. And they help to stop pathogens from attaching to the epithelial surfaces. Moving on to the IgDs. IgDs will be found on B cell surfaces, and they function as the B cell antigen receptor. Lastly, you have IgEs, which bind to mast cells and basophils. They trigger histamine release when we have inflammations, when we have allergies, and the major cause of allergic reactions. You should also know that the structure, so we have the IgMs are pentamers, meaning they can bind to 10 antigens at once. These guys are monomers, so they can bind to 3 antigens at once. These guys are dimers, so they can bind to 4. The IgDs are monomers, so they bind to 3. IgE monomer, it can bind to 3 antigens. So now we're going to talk about humoral immunity. Okay, so everything from this line over is humoral immunity, and everything before that was background knowledge you should be aware of. So in humoral immunity, we have the activation of B cells. So at this point, these 3 B cells, they are trained, they are ready to bind with antigens because they know how to attack them thanks to the maturation that happened right here. So the antigen picks one of these B cells to undergo clonal selection. So in this case, it's going to be this middle one right here. The antigen binds, and these guys that you see here, these are IgD antibodies. Because remember, we just went over, IgD antibodies are on the surface of B cells. So these IgD antibodies, they are trained, they are ready to fight foreign antigens, thanks to the maturation. So they proliferate, they form a clone, an identical copy, and then these 2 B cells are going to turn into plasma cells. And these plasma cells, you're going to get more plasma cells than memory cells. These plasma cells release IgM antibodies because we're talking about the primary response here in humoral immunity. The primary response will result in IgM secretions. You will also get 1 less memory cells than plasma cells, but you'll still get some memory cells from this. And this memory cell is primed. It's ready to go. It doesn't need to go through all of this again because it's ready to go to attack antigens because it already knows the pathway of how to do it thanks to the first primary response. Going on over here, so this is the memory B cell. We're just taking it over here. And this is known as the differentiation of B cells. So now once the same antigen binds to a memory B cell, it already knows how to respond. This is the secondary response. It's going to transform into even more plasma cells, which will secrete a whole lot of IgGs. Remember, these are the most abundant in the body. So it makes sense there's going to be a whole lot of IgGs. You're also going to get even more memory cells, which will be able to do the same thing and just increase the response you're able to get. All right, so the effect of this memory B cell, if you come on over here, this is the primary response, which is what you saw with this. But this line here is the secondary response, which is what you saw with this. The reason why it's stronger and it lasts longer is because more IgG antibodies get secreted in the secondary response than IgM antibodies in the primary response. So that explains this increase here. Another thing to note is that in humoral immunity, there is something known as active immunity and passive immunity. Actively, this is naturally required where you're exposed to a pathogen through an infection or some sort of natural contact with a pathogen. You can also do humoral immunity artificially. The biggest example of this is with a vaccine. Right, so you get a vaccine, you get a little bit of that virus or that bacteria injected in you to train your B cells to attack these foreign antigens to keep you healthy. Now, passive immunity, this you also have naturally acquired or artificially acquired. The naturally acquired passive immunity is when the antibodies are passed to the baby through the placenta, or to a baby with her who breastfeeds, right? Because IgA antibodies come from mother's milk, so that's how you passively naturally acquire IgA antibodies. You can also do it artificially, where you inject exogenous antibodies through a blood transfusion. Humoral immunity in a nutshell, this graph right here. When you have the antigen-antibody complex, which we just showed you with all the B cells, you go and you can do neutralization, which is shown here. You can do agglutination, which kind of just causes the cells to burst and to explode. You can do precipitation, and eventually this is going to trigger the complement pathway of activation like you saw with the innate immune system. And so this is eventually going to lead to cell lysis, and it will also influence and expand phagocytosis and cause even more inflammation. So now with all of this going on, you can see how this feeds into the innate immune system, because the innate immune system is all about phagocytosis with those macrophages. It's all about inflammation, right, with that chemotaxis that's going on with the mobilization of the neutrophils and then the macrophages. And then eventually you will lead to cell lysis. All right. Moving on to cellular immunity. Cellular immunity is all about CD4 cells, CD8 cells, and T cells. Right? Another rule of thumb to keep in mind is that B cells all deal with humoral immunity. T cells all deal with cellular immunity. Okay, so we start out with MHC proteins. What are MHC proteins? They're histocompatibility proteins. Histo meaning tissue, compatibility meaning match or no match. They consist of a peptide, a self-antigen, and a foreign antigen. And T lymphocytes can only bind to antigens presented to them by MHC proteins, because MHC proteins tell your cells that those cells are of you. You need an MHC protein on it to inform the cells around it that that cell is part of your own body. Now, what are antigen-presenting cells, aka APCs? The job of an APC is to engulf antigens and display them for other T cells to recognize them. APCs, they consist of dendritic cells, and dendritic cells engulf antigens and migrate to nearby lymphatics, so the nearby spleen, the nearby lymph nodes, to display antigens to T cells, which you can find in those lymphatics. You've got macrophages, and the macrophages presents the antigens to T cells to activate them. And you have B lymphocytes, which present these cells to the helper T cells. Going into a little more detail about MHC proteins. MHC proteins are divided into two classes, 1 and 2. Class 1 is on every single cell in the body except erythrocytes. These class 1 MHC proteins activate CD8 cells and inform the cytotoxic T cells to do their thing. And we'll get into that in a second. Class 2 MHC proteins are found on only cells that present antigens to CD4 cells, consisting of these three things right here: dendritic, macrophages, and B cells. This is the diagram of how T cells work. Okay, so you start out with your immature lymphocyte in the red bone marrow. You undergo maturation of T cells in the thymus. And then in the thymus, you create CD4 cells and you create CD8 cells. These are the different types of T cells. Now this is going to be activated when an antigen presenting cell, in this case it's just a dendritic cell, binds with a CD4 cell with a class II MHC protein displaying the antigen of the bacteria. Then you divide more and you create a memory CD4 cell. Kind of similar to what you saw with the B cells, but this is just creating a memory T cell. But then you keep going and you clone this T cell to make two identical copies with information from the APC. And then these will transform into CD4 cells will become either helper T cells or regulatory T cells. Coming on over here with the CD8 cells, they will be activated by an APC dendritic cell with a class I protein displaying the antigen, right? CD8, think class I. CD4, think class II. They're going to do the same thing. They're going to create a memory CD8 cell, so it's primed, it's ready to go. It doesn't have to do all of this again. And then you're going to, with the cell that's activated, you're going to clone it, do clonal selection with this information. And then you're going to divide even more, and then you're going to create cytotoxic T cells. Okay. And those cytotoxic T cells ultimately lead to cell lysis, to cell explosion. You eventually have to get from this in cellular immunity to a cytotoxic T cell. We're going to explain how to do that in just a second. Come on over here to these slides. So this picture you see right here is literally just a zoomed in picture of this. This slide over here is just showing you how it's done, zoomed in. So with the example of a CD4 cell, you have a class II MHC protein that displays the processed bacteria from the antigen. Remember, this dendritic cell is a macrophagy and it has already undergone phagocytosis. So it's already ate the bacteria and it's just displaying the fragmented part of the antigen to the CD4 cells to ensure full destruction. Okay. When the CD4 cell binds to the dendritic cell, you have its TCR and its CD4 binding to the MHC proteins here. Okay, so this CD4 protein is going to bind to the antigen, and the T cell receptor is going to bind to the MHC class II protein. And then you clone it. So you make a memory one, and then you create three that are going to become those regulatory T cells or those helper T cells. Now that's exactly what we're going to go into. What do helper T cells, what do cytotoxic T cells, what do regulatory T cells do? A helper T cell in humoral immunity, right? We're talking about humoral immunity here. In humoral immunity, the helper T cell will bind with the self-non-self complexes of a B cell that has encountered its antigen and that is displaying it on MHC2 on its surface. Okay, so this helper T cell is going to bind with a B cell, right? That B cell is going to have, this helper T cell is going to bind with this B cell because this B cell is going to have an MHC2 protein on it, displaying its antigen that it has encountered. And this is just going to help the B cell do its thing by releasing interleukins as co-stimulatory signals to complete B cell activation so that that B cell can then go and then turn into either IgM antibodies or IgG antibodies, depending on if it's a primary or secondary response. In cellular immunity, the helper T cell will bind to the dendritic cell so again, this is going from we're talking about the CD4 proteins, because remember, the CD4 proteins eventually have to become, or rather the CD8 cells that transform into helper Ts or regulatory Ts eventually have to become into cytotoxic T cells to completely destroy what you're trying to destroy. So the helper T cell binds to the dendritic cell, and the class 2 MHC protein is located on that dendritic cell. You have the CD4 protein, which is binding to the dendritic cell as well, and helper T helps express the co-stimulatory molecules when you bind to the CD8 cell, because the dendritic cell has class 1 MHC protein on it as well, because remember, anything that has class 1 has to have class 2 as well. So you have both. You have an MHC2 protein and you have a MHC1 protein on your dendritic cell. So when the CD8 cell binds to the dendritic cell, it binds to the class 1 MHC protein. And then now you've transformed this CD4 cell, and this CD4 cell is all done. Now you've fully activated a CD8 cell, and the CD8 cells become cytotoxic T cells after activation. And the cytotoxic T cells, as you can see through this pathway, they make a perforin pore, and they release granzymes into it, which just widens the hole and causes the bacteria to burst and to explode and to finally be out of the body and fully destroyed. What the regulatory T cells do is these three things: they dampen the immune response, help to prevent autoimmune responses, and they release inhibitory cytokines. And that is how the adaptive immune system works.